Hello everyone, this is Dr. Rasi and welcome back to the channel. Dr. Rasi explains. So today topics are nutrition, plant hormones, and plant genes and genetics. So it is a review for exam 4. So let's get started. So again, there are three main chapters. We will start with the first chapter, which is plant hormones or plant growth regulators. They are also called phytohormones. So again, because they are plant hormones, and like any other hormones, they are small organic molecules occur naturally in plants. They regulate, they are very important, obviously, and they regulate physiological growth processes such as flowering, germination, stem elongation, dormancy, fruit ripening, and gene expression, very importantly. So there are several plant hormones, um, from gibberellins, GA3, to ABA to ethylene. We are going to cover the major 8 of them, the 8 big ones, but really 2 4 5 biggest ones are auxins, cytokinins, ethylene, abscisic acid, and gibberellins. So you see here, um, again, you need to do your reading from the book in this chapter 27, and make sure that you do some deep reading really before you really studying, completing the studying in this chapter. So first we start with the auxins. As you see here in this table on the screen, you can see they all affect gene expression because there is a plus sign in front of all hormones. So gene expression is influenced by all of them. So that makes them really, really important. Okay. So some affect cell division rate positively, or cell expansion. We know already cell expansion also very important. Also direction of cell expansion, and again gene expression. So our first hormone is auxins. Auxins are the first hormone, that plant hormone, that's discovered, which promotes cell enlargement. Okay, they are produced at the shoot apical meristem. So they go and travel down, promote root initiation and flowering, xylem differentiation, and bending of the stems. We know from some experiments, and apical dominance, which is something that auxin can cause. Actually dominates the lower level, buds, so that they don't actually sprout because of auxin domination there. Okay, so very important hormone. Cytokinins is the second one, which promotes cell division. Unlike auxin, cytokinin actually made in roots, and they move up and involve in shoot formation, activate photosynthesis, remove apical dominance. You know, we just seen, which is caused by auxins, but cytokinins can remove that, and delay senescence, and or biological aging, which is senescence. Another big one is gibberellins, like GA3, which promote internode elongation. So that means plants get taller and taller because of gibberellins, or some plants are actually short um, because of absence of gibberellins, right? So they are both important, and they can also break dormancy. They can mobilize the enzymes in germination. They can actually cause flowering, fruit development, and then cause taller plants. Okay, again, if you have shorter plants, like dwarf plants, that means they are actually missing important gibberellins. Next one is abscisic acid, or also ABA, which inhibits or cause dormancy and stomata closure. Now, inhibit germination. So inhibit germination, that means it's caused dormancy in seeds. And it can also suppress flowering, growth cell elongation, abscission of leaf. So you can see a lot of negative effects caused by this important enzyme, this important hormone called ABA. Next one is ethylene. Ethylene is CaC2H4, which is another hormone that inhibits senescence, but very important, it's known for fruit ripening. Okay, it can also inhibit cell elongation. So next one is salicylic acid, which is SA, which promotes defense. We know it's actually plants use it in defense against bacteria. So that's why it's also a good compound for aspirin. Um, it's from bark of a willow tree. This aspirin comes from. It's a systemic acquired resistance, which is a plant immune response plants use it for. Carotenoids are also another newer hormones that they cause cell elongation, cell division, stress resistance. And then jasminoids, lastly, GA, regulates plant response to abiotic stress, plant defense, growth, photosynthesis, and gene transcription. So this is like a quick summary of this chapter. Our next chapter is plant nutrition and soils, a very, very important chapter. Plant nutrition, obviously, is directly related to human nutrition and animal nutrition. That makes it very important. So there are two groups of nutrients. They are called macro, MACRO, macronutrients, the big ones: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. And then there are micro, which are the trace elements like zinc, manganese, iron, molybdenum, nickel, boron. So copper are in this group, and also iodine. If plants are missing or low in these nutrients, they show deficiency symptoms. In your book, there are pictures of each deficiency. It can be like iron deficiency, yellowish, whitish leaf on more close to the top of the plant. Nitrogen deficiency, light green leaves, more lower part of the plant. Zinc, chlorosis and necrosis, again in the middle of the plant. Phosphorus, darker leaves in the lower part of the plant. Magnesium, yellowish colors, green veins in the lower part of the plant. Another important point is soil. Soils are very, very important, and they actually takes hundreds of years to develop soils. There are horizons like A, B, C, R, then from the top to bottom, and then there is O horizon, which is the very, very top, which is called organic matter. A is like 0.1 to 0.3 meter. That's also called topsoil. B is 0.3 to 1 meter. It's called subsoil. C is 1 to 1.3 meter, which is parent material. And then R is the very bottom, solid rocks. Okay, so there are different types of soil. Please do your reading on this. And then also, what are the acidic soils versus alkali soils, how they affect also plant nutrient availability. Okay, so the next chapter is our last chapter, which is genetics. We have another video on genetics you can also watch, just specifically on the genetics. Genetics start with vocabulary. Obviously, you need to know the terminology, start like learning, memorizing this basic vocabulary. And then you can actually build on basic Mendelian genetics. So always remember, phenotype equals genotype times environment. Genotype is the genetic makeup, that's the genes. This chapter, chapter 8. Environment is everything else we've seen in many other chapters, the environmental effects on plants. Every gene has a letter, like big letter is dominant, little letter is recessive. Mendelian genes have one gene ratios like 3 to 1, like flower colors in peas. They have incomplete dominance, like Snapdragon, like 1 to 1, 1 to 2 to 1 ratio, or codominance, like 1 to 1 ratio, or they could be dihybrid, like 9-3-3-1, 2 genes involved, or polygenic, which is also called QTL, which like height, eye color, seed mass, diabetes, diseases, these are all polygenic traits. So there are these experiments that are done by Mendel. That's actually very important. You need to review those, and then you need to know basic crossings and making Punnett squares. And so you know that trait, which is an inherited distinguished characteristics. genetics is heredity plus variation. Variation is very, very important. So it goes from DNA, chromosomes to genes. Alleles are just different forms of the same gene. Again, like blue eye color, green eye color, um, brown eye color. Okay, and so there is a little more information about the DNA. Please complete reading on this, and then keep studying, happy studying, and best of luck. And make sure that you actually subscribe to our channel to get more videos. And I'll see you in the next video.